So the previous class we had talked about the light dependent stage of photosynthesis, and just as a quick recap, it involves the light dependent stage involves chloroplasts where the pigments are arranged into two complexes, the photosystem 1 and the photosystem 2. Right, one thing I kind of skipped over the last time is that each of these photosystems absorb light in a slightly different wavelengths of the spectrum. So photosystem 1 absorbs wavelengths of light at around 700 nanometers. So I'll just write that down over here in black. So 700 nanometers is the wavelength of light photosystem 1 absorbs, and photosystem 2 absorbs light in the range of 680 nanometers. So not hugely different, but enough for various lights to be absorbed and utilized. Okay, so the pigments that are what make up the photosystems, and so those are what absorb the light, and that energy is used to split water, which then provides protons or hydrogen ions, and the oxygen that we know is produced, so to speak, as in photosynthesis. That's where it's coming from. It's actually coming from the splitting of water, and those protons are then later used to reduce carbon dioxide to carbohydrates in the light independent stage, which we'll be doing today. ATP is also produced, and that's also used to provide the energy to build the carbohydrates, again something we'll be doing today. The light dependent stage happens in two processes, and they're both happening simultaneously. One is cyclic photophosphorylation, where light is absorbed into photosystem 1 only, and because light is made up of photons which have energy, and so that energy is transferred to the electrons of the chlorophyll, and they become excited, they leave the chlorophyll molecules, and then they're picked up by a series of electron carriers, and the energy of the electron is used to convert ADP and inorganic phosphate to ATP, which is the photophosphorylation part. And because it's cyclic, the electron is returned to chlorophyll, and as I mentioned, only photosystem 1 is involved. Non-cyclic photophosphorylation involves both photosystem 1 and 2. Light is absorbed by photosystem 2, electrons gain energy, they leave the chlorophyll, they're picked up by an electron acceptor, and they move along the electron transport chain. ATP is produced. That electron then reaches photosystem 1. Light energy is absorbed by photosystem 1, electrons are excited, they're released, and then they're taken up by ADP. Now it's not as if the electron released by photosystem 1 is the one that it gained, is the same electron that it gained from photosystem 2. No. Photosystem 1 and 2, or 2 and 1 in this case, are releasing electrons at the same time. The difference is that the electron released by photosystem 1 is taken up by NADP, and the electron released by photosystem 2 is taken up by photosystem 1 to replace the one that it's lost. Now, two things are pending, the electron lost by photosystem 2, so you don't want it to have charges and lost electrons, because that will cause stability issues. And secondly, the NADP has gotten an electron but still needs hydrogen. So what's happening? Photolysis. So light energy is used to split up water, and that's how it happens. So H2O, that's water, light energy is split into a hydrogen ion or a proton, and it's taken up by NADP to produce reduced NADP that's going to come into play in the light independent stage. And then you've got all these OH ions. So as far as NADP is concerned, this is the entire equation. And as for the OH ions, the electrons are used to replenish the ones lost by photosystem 2, and the rest of the OH, 4 of these OH ions, they lose their electrons, and then they form oxygen, which is released by the leaves as a waste product or utilized within the plant by the cells for respiration, and some of the water is replenished, so to speak. So that was a recap of the light dependent stage. Any questions? All good?
Yes, sir.
I do hope you are keeping up with reviewing the text as we
Yeah. Yes, sir. Yeah.
yeah. So today we will be doing the light independent stage, right? And this utilizes the products of the light dependent stage, which are primarily reduced NADP and ATP, and the entire thing takes place in what is called the Calvin cycle. And you'll be surprised to know that glucose is not the primary end product. Right, so glucose is something that's made on the sides. So how does this work? So, right now, contrary to respiration, right, just to draw a parallel between the process of respiration, because you have the Krebs cycle over there, and the Krebs cycle was part of a series of reactions, the glycolysis and the link reaction. So the products of glycolysis went into the link reaction, and products of the link reaction entered the Krebs cycle, and so on. That's not how the Calvin cycle works, right? The Calvin cycle is working independently, right? The products of the light dependent stage are coming into play, but they're not the substrates of the Calvin cycle. Okay, so the Calvin cycle starts off with a compound, a 5-carbon compound called ribulose bisphosphate. So if I just write that down in points, right? Calvin cycle starts with a 5-carbon compound, right, which is called ribulose bis. Do you know what the bis means? It means two.
No. Oh.
Bi.
Okay.
Right, so there are two phosphates attached to the ribulose. Okay, and it has an accepted abbreviation of RUBP, the U being lowercase. That is an accepted abbreviation. Okay. Oh, by the way, the Calvin cycle, the light independent stage, takes place in the stroma. So the light dependent stage took place on the thylakoid membranes, right?
Yeah.
So this takes place at the thylakoid, right? Particularly the membranes of the chloroplasts, right? The Calvin cycle, the light independent stage, takes place in the stroma. So the ribulose bisphosphate is there in the chloroplast, and right, it is combined with atmospheric carbon dioxide. right? And this is a process called carbon fixation. So the carbon dioxide is fixed. Now that it was broken, it's fixed into the molecule. Okay? And what this does initially, it gives, what it gives, right? This is catalyzed by the enzyme ribulose bisphosphate carboxylase or oxygenase. So there's two of those, right? Ribulose bisphosphate carboxylase. These are fun names, huh? I remember back in the day when I was a bio student in A level, and we had students from the commerce side as well, and we used to show off. And they used to go like, "Oh, can you balance a sheet?" and whatever else terminologies they have, I don't remember. And we were like, "Okay, fine. Our terminologies are way cooler than balancing sheets." Okay?
Yeah, I agree.
Yeah, you cannot top stuff like ribulose bisphosphate carboxylase with a sheet balance.
Yeah.
Okay, so ribulose bisphosphate carboxylase or ribulose bisphosphate oxygenase, either of those two work, right? Is the enzyme that catalyzes this reaction, and it also has an right. It is called Rubisco. From writing that, oh no, it's all caps. All caps. Rubisco. Okay. Now what you get, because you're combining 5 carbon compound with carbon dioxide, it's 1 carbon, so you get a 6 carbon intermediate. It's an intermediate and it is unstable. So what happens here, it breaks up into 2 molecules of a 3 carbon compound, right? Something that we are familiar with. It is glycerate 3-phosphate. Glycerate 3-phosphate, right? And the 3 over here is just telling you the position of the phosphate group. And its abbreviation, known abbreviation, is GP. Now what happens to glycerate 3-phosphate? This is where the reduced NADP comes in, right? So the reduced NADP reduces, so it gives up its hydrogen, reduces the glycerate 3-phosphate, and we get a compound called glyceraldehyde 3-phosphate. This should be very familiar because we came across this compound in respiration. Glyceraldehyde 3-phosphate, right? Also known as GAP. Ring a bell? Faintly, maybe?
Yeah.
So you can go and check, right? It's there. And so GALP is the right main end product. Oh, and by the way, right, the energy for the carbon fixation and the reduction, right? So the energy for all these reactions, well, actually not all of them, right? In particular, the reduction of GP to GLP, right? Energy for reduction of GP to GALP, right, from ATP, the same ATP from the light dependent stage, right? The reduced NADP was also from the light dependent stage, and the ATP is also right from the light dependent stage. Now, as I was saying, the 3-carbon molecule, the GALP, is the main so to speak, the end product of the Calvin cycle, right? So to speak, because it is a cycle. So the most of the GALP to recover or reform the ribulose bisphosphate, most of it. So as I said it right here. Oh yeah. So glucose is actually made on the side. You would expect, oh well, the glucose is the end product, just like we had ATP coming at the end of oxidative phosphorylation, right, after the Krebs cycle. No, right? So if most of the GLP, the glyceraldehyde 3-phosphate, is being utilized to reform ribulose bisphosphate, then how do we end up with glucose? Well, before I move on, let's try to draw the Calvin cycle as a cycle. So you've got, let's change colors. You have ribulose bisphosphate, and then you've got CO2 coming in, right, and rubisco, and you get a 6-carbon intermediate that very quickly breaks down to 2 molecules of glycerate 3-phosphate. And then the glycerate 3-phosphate is converted, not a very nice circle, better slightly. You get 2 molecules of glyceraldehyde 3-phosphate, so GALP. And over here you have the reduced NADP, going back to regular NADP, so it's oxidizing, it gives up its hydrogen, and ATP is also utilized. And from here, right, now there are a bunch of other intermediates that happen because that are formed because, right, 2 3-carbon compounds don't automatically make up a 5-carbon molecule, right? And so, right, those intermediates are not mentioned, but right, the majority of the GALP goes back to forming ribulose bisphosphate, and some of it leaves the cycle to form glucose. That is the Calvin cycle. And the name of the scientist after whom the cycle is named is pretty cool, right? His name was Melvin Calvin. It's a cool name. Okay. So that is the Calvin cycle as a cycle. Now, how do we get the glucose? So we'll talk about that. Right? Is just to keep in mind that the light independent stage can occur in the light and the dark. So just because it's light independent doesn't mean that it occurs only in the dark without the light at night. No. So it occurs in the light and the dark. The only thing is that because it relies on the input of reduced NADP and ATP that is coming from the light dependent stage, right? It depends or relies on reduced NADP and ATP from the light dependent stage and so what will happen if the plant is kept in the dark for too long? Right, photosynthesis will stop. And why will it stop? Because no more glucose will be produced. Why? Because the light independent stage will stop. Well, that doesn't make sense. It doesn't need light. Why will it stop? Okay, independent.
Respiration will also continuously happen.
Well, respiration is not dependent on light. So yeah, right. It just depends on glucose being there. Right, so the light independent stage will stop, and the reason for that is this thing, because you've kept in the dark for too long, you are not getting the reduced NADP and ATP that's coming in from the light dependent stage, right?
Yeah.
So those compounds that are feeding into the Calvin cycle will be depleted. So reduced NADP and ATP depleted, and so the light independent stage stops. You're not getting any more glucose. Right, now obviously in nature plants are adapted to the changing periods of light and dark throughout the year, right? And they're adapted and they're very efficient at doing what they do, and so you don't see plants just dying off. I mean, plants lose their leaves, they get they rejuvenate in the spring, all of that, but they survive, they don't die off, because even the winter, they lose their leaves. So you say, oh, how's photosynthesis happening? How are they alive? Well, the tree is still alive. right? It's not dead. The roots are alive. Everything's alive. And again, that only happens in certain regions, right? In our countries, in the tropical countries and the equatorial countries, you don't see most trees don't lose their leaves during the winter. So plants are very efficient in the natural environment, right? You wouldn't normally have a situation where it's kept in the dark for too long, right? Whatever length the night is, they're longer during the winter. The plants are good with that, right? Okay, now how do we get everything that we do from GALP? That is the question. Right, so right, some of the GALP, and once I explain this, it is given in text obviously in your book. Some of the GALP is used in cellular respiration, and right, some of it is used to produce glucose. So let's put a heading over here. Some is fed into the respiratory pathway, some is used to form glucose, and this process is known as gluconeogenesis. Right, gluco meaning gluco, neo meaning new. So gluco is glucose, neo means new, and genesis means making. Right, making new glucose. And then that glucose may be converted into sucrose for transport, or it may be converted to starch for storage, right, or cellulose, right, to form new cells. That's not the only thing that's going on. Okay. The GALP, when it enters, the respiratory pathway, it can also be converted to amino acids. Amino acids have nitrogen, so plants get their nitrogen from the soil. Right, nitrates in this case, in the form of nitrates. For the amino acids. And then, right, as I said, if we go back, at this point between the GALP and going back to ribulose bisphosphate, there are certain other intermediates, right? So GALP is converted in the Calvin cycle to a 4-carbon intermediate which is combined with phosphates from the soil to produce nucleic acids. Nucleic acids. Right, so RNA and DNA. Actually. Then also in the cellular, in the respiratory pathway, GALP is converted to acetyl coenzyme A, right? So we've come across that. And this acetyl coenzyme A can then, be used to synthesize fatty acids and then lipids. Now for that, right, sometimes GP, so right, the glycerate 3-phosphate, the GP, is also utilized to make glycerol. But the main player over here is GLP. Right, and we know that glycerol is used in lipids. Right. So there is a flow chart in the text. If I just take you there, I can put that up on screen. Can you see that?
Yes, sir.
So it's a pretty good illustration.
Yeah.
The Calvin cycle running over here. Right, it mentions the 4-carbon intermediate that's formed after GLP and before ribulose bisphosphate. You get phosphates from the soil, you make nucleotides and the nucleic acids. The GLP and both the GP, they can be fed into cellular respiration. They have different, right? So it makes glucose cellular respiration, right? Utilizes glucose, right? Or it can lead to the formation of sucrose in this case, or other polysaccharides, so right, formation of glucose, the amino acids and then the proteins using nitrates in the soil, or the acetyl coenzyme A forming fatty acids and lipids, right? The glycerate phosphate is right also used to make glycerol, which is a component of lipids. Alright, so the flow chart of the entire thing, the explanation is given here in text. And if I just, so this is also a pretty neat diagram, right? And it is the full process of photosynthesis which occurs continuously in plants, right? So the left side is the light dependent stage, right? And you can see that they've given both the cyclic and the non-cyclic photophosphorylation, right? All of that happening, and then the reduced NADP and the ATP feeding into right, the Calvin cycle. And the Calvin cycle happening over here. ATP is utilized, right? Most of the GALP is going back to forming ribulose bisphosphate, and some of it is removed to make glucose, among other products. Now we have time, and so we can talk about limiting factors, right? What do you say?
Sure.
Okay, so I'll go back to the whiteboard. Limiting factors is, right, we've been doing this since our O level and IGCSE, so it's nothing special, so to speak, but it is part of the syllabus and we will review it. Over here they're just giving you a more technical understanding of why limiting factors limit the process of photosynthesis. Right, and so when we take a look at the entire process, we see that light is required, temperature is required, right? And carbon dioxide is used. You have the light dependent, the light independent stages, and you've got enzymes involved, and enzymes require certain temperature to work. And so we can understand, why are these things going to affect the process of photosynthesis? Right, because plants don't have the kind of homeostasis, and we haven't done that. That is actually the next chapter after this one. We should be beginning tomorrow. But right, nonetheless, homeostasis is something we have come to know in our earlier studies, and so plants don't have that kind of regulation that mammals do. Right, they do have some, but not the way mammals can maintain their internal temperatures. Right, plants do that through transpiration stuff, but still, right, so maintaining pH and maintaining temperature and other factors, and also, right, the carbon dioxide that they need and the light that they need is coming from is obtained externally, so they can't maintain light within themselves, they can't maintain carbon dioxide within themselves, right? They need far more carbon dioxide than what they produce as a waste product of respiration. So some of that is given off, some of that is utilized within the plant, but they need much more of it, and so they require, right, uptake from the atmosphere. Now, let's talk about light. Right, so light intensity affects chlorophyll because it gets excited and it releases its electrons, the cyclic and non-cyclic photophosphorylation, and it produces reduced NADP and ATP. So light affects chlorophyll, right, in the light dependent stage, obviously. Which affects the production of reduced NADP and ATP. Right, and so if there's not enough light, then you won't get enough of the light independent stage going on, and so you will have low levels of NADP, reduced NADP and ATP, and so less of that will be fed into the light independent stage, and therefore you will have less glucose being made, right? So, right, low light equals to low production, right, affects light independent state. So I'm just writing that, right, as hints, and we can understand, okay, fine, so it's going how is it going to affect the light independent stage, right? And so we're going to have lesser glucose being made, and therefore light becomes a limiting factor, right? And not just like the intensity of light, but the wavelength of light. So light becomes a limiting factor if there's not enough light, right? So both intensity and what wavelength of light. And this is perhaps especially important when plants are kept indoors in artificial light, right? Not so much, right? If they're out in the open, then they're in the natural environment, assuming that they're in the natural habitat as well, right? And so there shouldn't be any issues. But if you remove them from the natural habitat, or if they are not outdoors, then right, because you can't control the intensity of the sunlight, and it has all the wavelengths of the electromagnetic spectrum, right? The next limiting factor is carbon dioxide. Right, and this is something that directly affects the light independent stage. Right, the concentration of CO2. Right, now the atmospheric concentration of CO2 is about 0.4. Just a minute. Sorry, 0.04. Right, and so in the atmospheric CO2, and so in its natural environments, again, right, we're saying this from a perspective where we can artificially infuse CO2 into the environment the plant is growing in. If it is growing in a greenhouse or so, right, in natural conditions CO2 is a limiting factor, right, because it doesn't go higher than that normally. So artificially, right, providing higher concentrations CO2 levels will lead to the increase in photosynthesis. And this is something that, right, people who are cultivating plants, fruits and vegetables in greenhouses, they do that, right? So they infuse carbon dioxide to raise the concentration in the enclosed environment, and so, right, more carbon dioxide is present, so more photosynthesis, so the plant grows more, it has better fruit, and so better yield. Okay, and finally, temperature. Right, and so we know that enzymes are involved in photosynthesis. Right, we talked about Rubisco, and so enzymes are sensitive to temperature, and they have optimum temperatures at which they work. Right, and so, right, the process of photosynthesis therefore is affected by temperature. Right, raising the temperature would theoretically increase the rate of photosynthesis. Right, of course, we are talking about if you are in a region where the temperature is lower. Right, in our regions where the temperature reaches 45, even 50 degrees centigrade, you don't really need to raise the temperature higher, because you're just going to end up killing the plant. Right, enzymes will get denatured. So, right, the example they give in the book when they're talking about rate of photosynthesis, comparing carbon dioxide concentrations, light intensity, and temperatures, is 20 and 30 degrees centigrade, which is pretty cool for our area, right? So we obviously have average temperatures much higher than that. So, right, but yeah, right, that's the reason why temperature affects photosynthesis. So again, if I just take you to the book very quickly to show you that graph, that is sort of a summary of all the limiting factors. You can see this graph over here, and they've got rate of photosynthesis on the y-axis. Light intensity is increasing. You can see at 20 degrees and 0.1% carbon dioxide, which is still higher than what atmospheric concentrations are, right? And you can see it levels off pretty quickly. You raise the temperature, you can see higher activity. You keep the temperature the same, you raise the carbon dioxide concentration, still you get increase. And if you raise both the temperature and the carbon dioxide concentration, so with increasing light intensity, right, you get even higher rates of photosynthesis. This is right, so this is a graph where you've got 0.03, so that is atmospheric concentrations, and then you've got 0.13, that's obviously much higher, it's about a tenfold increase or so. And as you increase the light intensity, you can see, ooh, right, rate of photosynthesis like really shooting. Okay, and that is all about the limiting factor. You can read this paragraph about, you know, the limiting factors in real plants. I talked about it during the explanation, and that is the end of this chapter. So next class we'll be doing homeostasis. Any questions?
No, sir.
Are you sure?
Yeah.
Alright then, we'll review the content from the text, and I'll see you next time, inshallah.
Inshallah.